Hello, my name is Laura Milko, and I represent the ClinGen Clinical Domain Working Group, or CDWG Oversight Committee. This presentation is intended to provide an overview of the ClinGen processes and recommendations for establishing expert panels for both gene curation and variant curation. First, I'd like to put the expert curation groups in the context of ClinGen's overarching goal to build a centralized and freely accessible resource that defines the clinical relevance of genes and variants for use in precision medicine and research. In addition to encouraging sharing of genetic and phenotypic data and establishing an informatics infrastructure to share our work, ClinGen has also developed evidence-based frameworks to support the exchange of expert knowledge about gene disease validity, variant pathogenicity, dosage sensitivity, and clinical actionability. We believe that engaging a global community of international experts and biocurators is the cornerstone of our mission to curate the clinical genome in an equitable and sustainable way. I'm very happy to have this opportunity to describe the architecture and operations of ClinGen's expert curation ecosystem and the immense collaborative effort of the many individuals worldwide who have contributed to it. The expert curation groups use three acronyms that I'll say frequently, so I want to familiarize you with these. At the broadest level, the curation ecosystem is organized by overarching clinical domains, such as cardiovascular disease, hereditary cancer, neurodevelopmental disorders, and so forth. Each clinical domain is led by an umbrella group called a Clinical Domain Working Group, or CDWG. CDWG members include domain content experts, coordinators, and biocurators of affiliated gene curation expert panels, or GCEPs, and variant curation expert panels, or VCEPs, performing curation activities in their broad clinical domains. I'll use the term expert panels generically when I'm referring to both variant curation and gene curation expert panels. The CDWGs are intended to cover all the major clinical domains, similar to those proposed for competencies for medical geneticists in this commentary by Korf, Iron, and Watson. You can see on the timeline at the bottom of this slide that we began establishing the first CDWGs in late 2013 through early 2015, and our process is described in the paper that's cited at the bottom of the page in Genetics in Medicine from 2019. As ClinGen has continued to expand, we've developed new CDWGs, and you can see on the timeline those that we've prioritized for future development. So we're currently on schedule as we're launching the neurodegenerative CDWG currently and its affiliated GCEPs and VCEPs, beginning with the Parkinson's disease GCEP. The CDWGs have a hierarchical organization via the leadership of the CDWG chair or chairs and an executive committee that is intended to be broadly representative of the clinical domain across key subdomains, such as clinical, research, and molecular laboratory expertise, and balanced demographics, for instance, the ratio of males and females, the level of seniority, geographic location, etc. Affiliated GCEPs and VCEPs, represented by the blue and purple circles on the slide, generally form under the guidance of a CDWG and are often led by members of the CDWG's executive committee and other community members who are invited to participate in the expert panel in their particular area of expertise. Here, I want to highlight the tremendous work of the coordinators of the CDWG and expert panels, as well as the biocurators, in facilitating the accomplishments of their expert curation groups. Though they are represented at the base of the pyramid and they do provide its foundation, they are active throughout the entirety of the CDWG and the expert panels and are frequently the driving forces behind the successes of the groups, from assembling the group for meetings, assigning and participating in curation and review activities, authoring manuscripts, and much more. Among the tasks of the CDWGs are to provide leadership and structure under which the expert panels can thrive, and to facilitate awareness of the numerous ClinGen resources that have been developed, such as educational and training resources to build effective workforces, curation frameworks and interfaces to perform and disseminate systematic and evidence-based curation, and also to integrate the expert panels into the larger ClinGen consortium so they can participate in and benefit from efforts to provide trained volunteer biocurators, outreach activities to expand their networks, and computer-assisted annotation efforts. I should note that occasionally some expert panels simply do not fit under an existing CDWG. For example, if they're broadly syndromic or if the overarching CDWG has not yet been established. In this case, the CDWG Oversight Committee stands in to fill the role of the CDWG. The CDWG Oversight Committee, of which I am a member, is co-chaired by principal investigators from each of the 3 grants, Jonathan Berg, Heidi Rehm, and Sharon Plan, and is also composed of leadership and key representatives from several existing CDWGs and expert panels and other ClinGen working groups that have frequent interaction with the expert curation ecosystem, such as the Sequence Variant Interpretation, or SVI working group, the Gene Curation Working Group, and the Education, Coordination, and Training Working Group, and also the NIH's National Human Genome Research Institute. It's important to have this cross-representation since the goals of the CDWG Oversight Committee are broad and overarching across the expert curation ecosystem. They are to set priorities for future CDWG and expert panel development, to promote harmonization and standardization of curation activities, to facilitate interaction between the expert curation groups and the larger ClinGen consortium, to provide an orientation and materials for expert curation groups, to inform them about the roles and responsibilities that they will be undertaking, and just provide general information about the consortium itself and the processes they will be implementing, to provide a point of contact for new groups who wish to form expert panels and use the ClinGen resources, and also to conduct outreach and foster relationships within the larger clinical genomics community to accomplish our mutual objectives. Now I'd like to turn our attention to the specific process for establishing gene curation expert panels, or GCEPs. GCEPs are charged with determining the strength of the evidence that supports a causal relationship between a particular gene and a disease of interest. Once established and approved by the CDWG Oversight Committee, GCEPs use the ClinGen Clinical Validity Framework, the Gene Curation Standard Operating Procedures, and the Gene Curation Interface, or GCI, to assess the strength of evidence supporting a gene disease association. Trained biocurators curate primary literature using the ClinGen semi-quantitative framework and scoring system to provide a comprehensive summary of the evidence for review and for a final classification decision by the experts. The GCEP approval process requires the completion of a stepwise process to define the composition of the group and its scope of work, and familiarize its members with the workflow, processes, and the gene curation interface. Groups must submit their Step 1 application to the CDWG Oversight for approval prior to initiating Step 2. On average, it takes about 3 months from the beginning of this process to approval by the CDWG Oversight Committee. After approval, the GCEP begins ongoing curation and in the long term ensures that their curations remain up to date and that discrepancies are resolved in a timely manner. The first step is to identify the group composition. Broad representation of the community of experts is strongly encouraged to ensure that the breadth of the expert opinions in the field is reflected and to mitigate issues with conflict of interest. Specifically, a GCEP must include at least 3 independent content experts from different institutions, though this is simply a minimum number and most GCEPs include more experts. Ideally, at least 1 member would have expertise in the ClinGen Clinical Validity Framework, and if the GCEP falls under an existing CDWG, a ClinGen staff curator is often embedded as a consultant for a small amount of effort. For GCEPs without an embedded ClinGen staff member, there will be training and periodic interactions with the CDWG Oversight Committee and the Gene Curation Working Group. The chair or co-chairs of the GCEP should be well-regarded experts in the field with invested interest in the success of the group. As mentioned previously, coordinators are an integral part of an expert panel as it is their role to assist the chair and facilitate the work of the biocurators and experts. Previous experience has shown to us that staff scientists, clinicians, and other people who are familiar with the focus area and its experts are often junior experts themselves, are often exemplary coordinators. However, for the coordinators to be successful, they must be empowered by their chair to assign and follow up with the activities of the members. A diversity of scientists and clinician experts is critically important to be able to properly review and interpret the relevant genetic and functional evidence. Expert diversity is necessary at various levels, particularly in terms of adequate representation of areas of clinical, diagnostic, and basic research expertise. Biocurators, much like coordinators, are generally staff scientists and clinicians with backgrounds in fields such as biochemistry, molecular genetics, or population genetics, and who may or may not be experts in the gene or disease of interest. New biocurators need not have had previous experience with biocuration, as ClinGen has developed a variety of materials to train and educate biocurators in the ClinGen gene curation processes. Once GCEP membership is established, the next stage is to decide on the scope of work of the GCEP, which includes defining the disease area or areas of focus and developing the gene lists being addressed. Gene lists vary widely, but generally speaking, starting with lists of genes that fall on genetic testing panels is a good place to start. It is expected that the GCEP will utilize ClinGen lumping and splitting guidance during pre-curation, which addresses an issue that is critical to gene curation, to define specifically what disease entity or gene is being curated and classified. Focus should generally be on the canonical disease, so splitting into multiple phenotypes should be avoided, and GCEPs will use the gene tracker to enter their pre-curation information. Both of these topics are covered in more detail in the education and training materials. Finally, I should mention that for GCEPs that eventually want to spin out a variant curation expert panel as well, it's important to include all the genes in which you eventually want to curate variants. An unavoidable consequence of engaging top experts in this work is the potential for conflicts of interest and competing interests, as many experts are actively working on concepts that have significant overlap with the mission of the CDWG. In doing so, GCEP members will be expected to self-manage and declare conflict of interest on a ClinGen survey that will be sent out by their coordinator. Expert panels and their overarching CDWGs are required to identify any conflicts of interest to ensure that members with academic or financial conflicts do not serve as the sole arbiter of gene or variant classifications for which they may have a biased perspective. So, for example, if an individual published the first paper to implicate a gene in a disease, or if they're a lab director or employee of a commercial laboratory. No special measures are needed if there's group consensus on a gene classification. Otherwise, those with relevant COI should refrain from decision-making in that case. Expert panels are also asked to adopt a collegial culture and to discuss timing of individual publications that may intersect with the interests of their group to avoid diminishing the impact of the collective ClinGen products. Before submitting the final application, the GCEP leadership will attest to some important conditions, including that the GCI will be used for all gene curation and classifications, the ClinGen publication policy will be reviewed, and a manuscript concept sheet will be submitted to the NHGRI and the ClinGen Steering Committee before the group prepares a publication for submission. And, according to our mandate as a freely accessible resource funded by the NIH, all curations completed by the GCEP will be made publicly available through the ClinGen website upon completion. Finally, that the ClinGen gene disease validity re-curation process has been reviewed and is acceptable to the GCEP. GCEPs also need to choose the process they will follow for performing gene curation and classification after they are approved. There are several ClinGen approved approaches listed here, and materials are available from GCEPs that have already completed the process to assist with this decision. At this point, the GCEP is ready to submit their application for review and approval by the CDWG Oversight Committee. Speaking of GCEPs that have completed the process, I want to showcase that there are 35 GCEPs currently in various stages of becoming an established GCEP, and also those that are currently curating their gene lists or recurating their outdated classifications. To date, there are 1,220 approved gene disease curations posted on the ClinGen website, and hundreds more gene disease pairs queued up to make their way through the gene curation interface and expert curation and approval pipeline. Now I'd like to turn our attention to the specific process for establishing variant curation expert panels, or VCEPs. VCEPs are the variant curation counterparts to GCEPs and follow a similar, rigorous, stepwise process for approval by the CDWG Oversight Committee to submit to ClinVar at the 3-star variant curation expert panel level. Because all ClinGen VCEP variant classifications are automatically recognized by the U.S. Food and Drug Administration FDA-recognized human variant database, all ClinGen VCEPs must follow an FDA-specified process for formation, approval, curation, assessment, and publication of variant classifications of pathogenicity. VCEPs must prepare submission materials for review and approval at the end of each step of the 4-step application process shown here, including approval by the CDWG Oversight Committee and approval of the ACMG AMP rule specifications by the SVI VCEP Review Committee. After final step 4 approval at the end of the process, VCEPs are ClinGen approved to submit variants to ClinVar with 3-star FDA-recognized status. I'd like to note that a companion guide to submitting a VCEP application, called the VCEP Protocol, serves as a detailed manual for completing the steps of the VCEP approval process. I'm going to step through some of the highlights, but you'll be able to access all of this information in more detail and with links to all the resources you need to complete these steps in the VCEP Protocol. We highly recommend that VCEP chair or chairs, coordinator, and any other key personnel read the VCEP protocol thoroughly before beginning the application process, and we also encourage you to communicate frequently with the CDWG Oversight Committee about any questions that arise during this process. Our recommendations about the types of expertise for VCEP membership is similar to that of the GCEPs, including individuals with diverse areas of expertise, including medical professionals caring for patients relevant to the disease gene in question, medical geneticists when the diseases span multiple organ systems, and basic science researchers with expertise relevant to the disease, gene, functional assays, and statistical analyses. For VCEPs, clinical laboratory diagnosticians and/or molecular pathologists who regularly use the ACMG AMP guidelines to classify variants and/or review variants during clinical laboratory case sign-out are particularly important, since a primary goal of the VCEPs is to specify the ACMG AMP guidelines for specific use in curating and interpreting variants in their genes of interest. In terms of biocurators, they are often genetic counselors and other individuals who are familiar with the ACMG AMP guidelines. In terms of scope of work for VCEPs, most focus on a gene or a set of genes associated with a single condition or related conditions and the curation of variants in those genes. Genes should be well characterized with a strong or definitive disease association and a known clinical impact. VCEP should begin with one or several genes or gene families that are the highest contributors to disease and/or the target of therapies or clinical trials within their area of focus for variant curation, and then steadily enlarge the scope of their project over time. The scope should include known gold standard variants for use in pilot testing. The VCEP conflict of interest process is the same as it is for the GCEP, so I'll skip over the slide this time. So at this point, the VCEP is ready to submit completed Step 1 application materials to the CDWG Oversight Committee for review and approval before beginning Step 2 to develop specified ACMG AMP variant classification rules. One of the main tasks of the VCEPs is to develop disease gene specifications to the ACMG AMP rules, and then determine how those specified rules can be combined to classify sequence variants according to the 5 Mendelian classification criteria: pathogenic, likely pathogenic, variants of uncertain significance, likely benign, and benign. There are a number of resources available for VCEPs to refer to during this process, including general recommendations on the SVI webpage for using the ACMG AMP criteria that are intended to provide consistency in usage and transparency in classification rationale. There are also posted specifications for all the currently approved VCEPs, and new VCEPs are strongly recommended to review the work of these VCEPs and utilize already approved specifications that may be relevant to their group. A recently updated sequence variant curation SOP also provides guidance on variant classification using ClinGen approved processes and tools, as well as additional resources that have been identified as valid sources of evidence by the SVI. VCEPs may proceed with rule specifications using one or a combination of the following processes. Approach 1 is to subdivide the VCEP and assign a category from each of the ACMG and AMP guidelines to each subgroup for discussion, and then the subgroups will then bring their proposed specifications back to the larger group for feedback and final consensus approval. Or approach 2 is simply to develop and approve the specifications to the guidelines all within the full VCEP. After the VCEP completes a draft of their ACMG AMP specifications using a template and instructions provided by the SVI, they submit it to the CDWG Oversight Committee and schedule a time to present and discuss their specifications with the SVI VCEP Review Committee for written feedback. Once the SVI approves the VCEP's draft specifications, the VCEP begins their pilot process. Step 3 is the pilot test of the draft variant classification rules with known variants using the variant curation interface. The VCEP should test at least 10 to 12 path or likely path variants, 10 to 12 benign or likely benign variants, and 10 to 12 variants of uncertain significance or those with conflicting interpretations in ClinVar. It's important to use variants with a variety of different evidence types for the genes to test all relevant criteria codes, after which the VCEP should then refine their rule specifications and then curate all their pilot variants using their final specifications to confirm validation. After completing the pilot, the VCEP sends their pilot results and final refined specifications to the CDWG Oversight Committee for review. Please note that if any ACMG or AMP specifications have been changed, they will need re-review by the SVI VCEP Review Committee. Once they are approved, it's time to move on to Step 4 and final VCEP approval. Now, VCEPs will define their plans for ongoing variant curation review and discrepancy resolution from one of the 2 ClinGen approved processes listed here, or often a combination of the 2 processes is used, depending on the complexity of the variant type. The VCEP also defines its meeting schedule to review, discuss, and resolve differences in interpretations. The final variant classification requires at least 3 core approval members, defined as those who regularly use the ACMG AMP guidelines to classify and/or review variants during clinical laboratory case sign-out. Then, a summary of approved variants is sent to the entire VCEP to ensure that any members absent from a call have an opportunity to review each variant. For clinically relevant variants, it's crucial to keep interpretations up to date and prioritize conflicting assertions that have been submitted to ClinVar after the VCEP submission. Therefore, the ClinGen approved schedule for recuration is shown on the slide, and the VCEP must attest to it for approval. To facilitate external review and understanding by end users, all finalized approved variant classifications must be accompanied by brief yet comprehensive evidence summaries. As you can see from the color-coded example at the bottom of the slide, the evidence summaries must include the condition for which the variant is being assessed, the evidence category codes applied and the reasons for applying each code, the pathogenicity classification, and a detailed rationale of any exceptions to the specified rules. For Section 4H, you'll need to ensure that anyone who plans to curate, both biocurators and experts, have completed and attested to variant curation training and are enrolled in the ClinGen Community Curation Database. Anyone who has completed this process is eligible to train new biocurators, so you'll want to designate 2 or 3 biocurator trainers for this on your application. As I just mentioned earlier in the variant curation and review section, you'll also need to identify a minimum of 3 core approval members who routinely use the ACMG AMP guidelines to classify and/or review variants during clinical laboratory case sign-out. Since you need at least 3 core approval members for variant curation classification approval, it's helpful if you can include an additional 1 or 2 members in the VCEP with this experience so they can alternate if needed. Finally, according to the ClinGen publication policy, since we are a freely accessible resource, all ClinGen expert panels must attest to disseminate their curation results immediately upon completion of expert review of each variant, along with all variant level evidence used in its curation. Generally, VCEPs publish their rule specifications in a peer-reviewed journal, and a draft of this publication must be sent to the SVI for review prior to submission. Within a month of submission, we'd like you to pre-publish your work to medRxiv or another pre-publication server. After Step 4 is complete, the entire VCEP application is ready to be sent to the CDWG Oversight Committee for final review and approval. VCEP leaders present a brief summary of their Step 4 plans on a CDWG Oversight Committee call, and then, upon approval, the fully fledged ClinGen Variant Curation Expert Panel finalizes their first set of 3-star, expertly curated, FDA-recognized variants for upload to the ClinGen Evidence Repository and ClinVar within 30 days. I'm proud to highlight the 42 VCEPs who are currently in the various steps of this process and the 18 publications from and about these VCEPs. Currently, there are 12,242 expertly curated variant classifications in ClinVar, but this number has been growing rapidly as more VCEPs enter the pipeline and complete the process. As of today, there are 2 VCEPs in Step 1, which moves pretty quickly, 19 VCEPs in Step 2, 8 VCEPs in Step 3, and 13 ClinGen approved 3-star variant curation expert panels. I'm happy to take any questions you may have now and answer any follow-up questions you may think of later. I've copied a link to visit for more information, as well as my email address and the email address of the CDWG Oversight Committee, where questions are answered pretty promptly by myself or one of my colleagues. Thank you very much for listening to my presentation.